What is photophosphorylation? What is photophosphorylation? You guys remember phosphorylation, right? Phosphorylation is the process where we add phosphate to a molecule, isn't it? Phosphorylation, as what we learned, is the process where we add addition of phosphate group to a molecule. That is phosphorylation, isn't it? Okay, when we say photophosphorylation, photo means using the light energy. Using the light energy. So photophosphorylation means a process where we add phosphate group to a molecule using the energy derived from the light energy. Okay, so that is actually the reaction occurs in the photosynthesis, because photosynthesis, we are producing ATP with the light energy as the source of the energy. So that's why photophosphorylation occurring in this process. What is photophosphorylation? So photophosphorylation specifically referring to the formation of ATP. Okay, how ATP is formed? Of course, with the addition of inorganic phosphate group to the ADP, isn't it? Okay, so that's formation of ATP lah. Addition of the inorganic phosphate to the ADP. But why it is called photophosphorylation? Because this process using light energy, okay, using the light energy as the source of energy, and this process occurs in the photosynthesis. Okay, photophosphorylation, remember what is that? That is a reaction to form ATP. How to form ATP? By adding phosphate group to the ADP, adenine diphosphate, using the light energy as the source of energy in photosynthesis. Okay, so that is photophosphorylation. And then photophosphorylation, there's two types. One is non-cyclic. Non-cyclic, tidak kita semula. Tidak kitar. Cyclic means kitaran. Another one is cyclic photophosphorylation. Another one is kitaran. Okay, this kitaran, cyclic and non-cyclic, actually referring to the electrons flow. Okay, so we are going to cover the first one, non-cyclic photophosphorylation. Non-cyclic means we are not cycling the electrons. Okay, the electron flow is linear. Okay, the electron flow is linear, not cyclic electron flow. So this is non-cyclic photophosphorylation. What does it mean by non-cyclic photophosphorylation? That means we are producing ATP. We are producing ATP by adding the phosphate group to the adenosine diphosphate to form ATP using the light energy with the involvement of linear electron flow. Okay, with the involvement of linear electron flow, means the electron is not cycle. It's not in cycle. Okay, so this is the overall of cyclic photophosphorylation. Okay, where is photophosphorylation here? Here at step 5. Okay, you see step 5? Okay, this is where we got the ATP. So this is where we say this is photophosphorylation. Why photophosphorylation? Because we are producing ATP at step 5 using the energy derived from the sunlight. Okay, which is this one. Okay, this one at step 1 here, I circle here, so we can see there's a light energy involved in this process. So at step 5 we got the ATP. So this is what we say photophosphorylation, production of ATP using the light energy in the whole process. Okay, and then you see the arrow here, all these arrow here, the black color arrow. Okay, the black color arrow here showing the energy transfer. You see the gold arrow. Okay, the gold arrow here showing electron transfer. Okay, showing electron transfer. All the gold arrow here showing electron transfer. Okay, and the electron flow linearly. They are not being cycled. They are not in cycle. They flow linearly. Okay, the gold arrow showing electron transfer linearly. Right. So let's go to the first one. Okay. Once I speak about light reactions, we start with PS2 first. Okay, we start with PS2 first. PS2 or photosystem 2 is the first photosystem function in light reactions. Why it is called photosystem 2? Because they are discovered later than the photosystem 1. Okay, but it occurs during light reactions. After the photosystem 2 function, then only followed by the photosystem 1 in these reactions. Okay, we start from the first step. This one, you see here, this is photosystem 2. There are lots of photosynthetic pigments on the light harvesting complex, and then at the reaction center complex at the center here, we have the chlorophyll A molecule called the P680, and then we have the electrons, and then we have the primary electron acceptor. Okay, the first step. What happened at the first step? So a photon of light strikes a pigment molecule in the light harvesting complex of photosystem 2 or PS2, boosting one of its electrons to a higher energy level, or the higher energy level is called excited state. Okay, so you see here, step number 1, you see light striking. So the light is absorbed by this pigment molecule, this photosynthetic molecule. So when this photosynthetic molecule absorbs the light, what happens is that the electrons of the chlorophyll, or this one is not necessarily chlorophyll. This could be chlorophyll A, this could be chlorophyll B, this could be carotenoids. Okay. So when the light striking to the pigment, so the pigment will absorb the light, and then the electrons, okay, remember the electrons of the pigment will be raised to higher energy level or excited state. Okay, so that's what happened lah. Okay, next one. So when these electrons are raised at its excited state, the excited state or the higher energy level is unstable. So because it is unstable, so the electron will be immediately dropped back. Okay, so like this one is the pigments. Okay, so the electrons will be raised at the higher energy level or the excited state, but then immediately the electron will be dropped back. Okay, so as this electron fall back to its ground state, an electron in a nearby pigment molecule is simultaneously raised to an excited state. Okay, so like for example, this pigment molecule, they have electrons. Once they absorb the light, the electron will be raised to higher energy level or excited state. Once it raised to excited state, then it will fall back. Okay, and then it will fall back to the ground state, and that will cause the pigment molecule nearby it or next to it to raise its electron to a higher energy level to the excited state, and then fall back to the ground state, and then the energy will be relayed to the next pigment, and then that will occur continuously. Okay, the energy will be relayed from the next pigment to another. Okay, so the process will continue with the energy, okay, with the energy being transferred to other pigment molecules until it reaches the P680 pair of chlorophyll A molecules in the PS2 reaction center complex. So this is actually referring to the black arrow here. You see the black arrow here? So this black arrow showing the energy is being transferred. Don't say electron transferred. No, electron is not transferred. The electrons for each pigment is raised to excited state, but then the electron will fall back to the ground state. Okay, when the electron are fall back to the ground state, energy will be transferred to the next pigment. Okay, energy, not the electron. The energy will transfer to the next pigment, and then the next pigment, causing the electron of the next pigment to raise to the excited state and then fall back, and then that will cause the energy to transfer to the next pigment. Okay, and then this process continue. So the energy will transfer from one molecule to another molecule, to another molecule, to another molecule, to another molecule, until the energy reach the reaction center chlorophyll. Okay, and in this case, the reaction center chlorophyll is called P680. Right, and then P680 is of course chlorophyll A, not chlorophyll B, not carotenoids. That's why P680. That's why the chlorophyll A, we say the dominant photosynthetic pigment. Okay, because they are the photosynthetic pigment in the reaction center complex. Okay, now once the energy reached the P680, what happened to the P680? Okay, what happened to the P680 when the energy reached the P680? Okay, you see in this diagram, you see the electrons are being raised at the excited state. Okay, so that's what happened for the P680. Once the P680 receive the energy, the electrons of the P680 also will be raised to excited state. Okay, that's what happened to the P680. Once they got the energy, then the electrons of the P680 will be raised to the excited state. Okay, so the energy will excite an electron in the P680 pair of chlorophyll A molecules to a higher energy state or the excited state. Okay, but then the electrons, for this one, the electron is not immediately dropped back. Why? Because the electron is accepted by the primary electron acceptor. Okay, in this case, for step 2, supposedly the electron will fall back just like the others, but that's not the case for this one. The electron raised to a higher energy state, excited state, but it is then accepted by the primary electron acceptor. So that's what happened. This energized electron is transferred from the excited P680 to the primary electron acceptor. Okay, so the primary electron acceptor, this molecule will accept the electrons and become reduced. Okay, this primary electron acceptor will accept the electron and become reduced. Okay, and then the P680 now, they are missing the electron lah, because the electron already accepted by the primary electron acceptor. So the P680, once they are oxidized, they are missing the electrons, right? They are missing the electrons. That's why they are. That means they are oxidized, isn't it? So when they miss an electron, immediately we call this chlorophyll as P680 plus. Okay, so P680 plus. Okay, whenever you see P680 plus, that means it is in oxidized state. Means it's already missing the electron, missing an electron. Why the electron is missing? Because the electron just now is raised to excited state and then being accepted by the primary electron acceptor. So there is an electron hole. There is an electron hole in the P680 plus. That means they are missing electrons. Okay. Okay, next, because they have now missing an electron, this electron needs to be refilled. This electron hole, okay, needs to be refilled, need to be replaced. Okay, this electron needs to be replaced so that P680 plus will be reduced back to P680. Okay, I repeat, P680 plus now missing an electron. That means there's an electron hole that needs to be refilled. Okay. Once this electron is replaced, then the P680 plus will be reduced back to P680. Okay. Now the question is, how is the electron that has been donated to the primary electron acceptor replaced? Okay, because now we have P680 plus. P680 plus missing an electron, and that's the electron hole that needs to be refilled, needs to be replaced. The electron needs to be replaced. So where can we get the electrons? So now there comes the H2O, water. Okay, now only the water or H2O molecule will comes into the picture. So water is required in light reactions here, okay, to refill the electrons, to refill or replace the electrons missing in the P680 plus. Okay, so you see in this diagram here, the H2O, it will be split. Okay, there's a process called photosynthesis of water will occur after this. So the water will be split into protons and also electrons and also oxygen. And then the electrons that is produced from the splitting of the water then will refill or replace the electron missing in the P680 plus. Okay, so that's what happened. So this occurs through the photolysis of water. This process is called photolysis of water. Okay, what is photolysis of water or water splitting? So an enzyme catalyzes the photolysis of a water molecule into 2 electron, 2 proton, and 1 oxygen atom. Okay, and 1 oxygen atom. One photolysis of water molecule, okay, a photolysis of 1 water molecule will result 2 electrons, 2 proton, and one oxygen atom. So this is the formula. Water split into half oxygen here, actually one oxygen atom. Okay, and then 2 proton and also 2 electron. Okay, you can see here there's 2 electron here, isn't it? So this electron is the one that will go and replace the missing electron in the P680 plus. Okay, and then the P680 plus, once receive the electrons from this, will then reduce back to P680. Okay, P680 plus, then reduce back to P680 to allow the next, to allow receiving the energy from the next transfer of energy. Okay, so the electrons from the water molecule splitting are transferred one by one to the P680 plus pair, each electron replacing one transferred to the primary electron acceptor. So the electron hole of P680 plus is filled. Okay. Okay, so when the electron hole of P680 plus is filled, so the P680 will be reduced back to P680. Okay. All right, and then what happened to this oxygen? Now, a photolysis of one water molecule will produce one oxygen atom, isn't it? Okay, so in order for us to produce one oxygen molecule, okay, in order for us to produce one oxygen molecule, we need 2 oxygen atoms, isn't it? Because oxygen molecule consisting of 2 oxygen atoms, O2. So we need 2 oxygen atoms. That means to produce one oxygen molecule, we need twice photolysis of water. Okay, we need twice photolysis of water because one photolysis of water will produce one oxygen atom. So the oxygen atom will combine with another oxygen atom generated by the splitting of another water molecule. Then only we can get one oxygen molecule. Okay, one oxygen molecule, and then this oxygen molecule is the one that will be released by the green plants, by the photoaltograph. So that's why the green plants, they during photosynthesis they release oxygen. Okay, that's why they release oxygen, because of these reactions. Okay, so twice photolysis of water will produce one oxygen molecule, and that will be released from the green plants through the stomata. Okay, and then the proton here that is produced here also will be released into the thylakoid space. Okay, that's all. So we have covered step 1 and step 2 and then step 3. Okay, step 1, the light energy striking absorbed by the photosynthetic pigments, electron of the pigment will raise to excited state, and then the electron will be immediately dropped back to the ground state, and that will relay energy to the next pigment, and the energy will be transferred from one pigment molecule to another until the energy reach the P680. Once the energy reach the P680, that will raise the electrons in the P680. The electrons will be raised to a higher energy state and will be accepted by the primary electron acceptor. P680 now oxidized to P680 plus. P680 plus now missing an electron, having electron hole that needs to be refilled. So where the electrons come from to refill the electron hole, so the electrons come from the splitting of water or the process of photolysis of water, where the water will be split into 2 electrons, 2 proton and 1 oxygen atom, and then the electrons produced from the photolysis water will one by one transferred to fill in the electron hole of the P680 plus, and the P680 plus can then reduce back to P680 and to receive another cycle, another transfer of energy. Okay, now that's settled. PS settled. Now, what happened to the electrons that is being accepted by the primary electron acceptor? So we go to the next process that is involving an electron transport chain. This one, step 4. Okay, step 4 here, you see this diagram. This is an electron transport chain. That means the electron is being transferred from one electron carrier to another. So in this electron transport chain, we have 3 members. We have PQ or plastoquinone. And then the second one, we have the cytochrome complex. And then the third member is the PC or plastocyanine. Okay, so this electron from the primary electron acceptor will be transferred to the plastoquinone, and the plastoquinone plus to cytochrome complex, and then cytochrome complex plus the electron to the plastocyanine. Okay, and then what's special about this electron transport chain is that during this electron transfer, ATP will be produced. So this is what we say photophosphorylation, because we are producing ATP using the light energy derived from using the energy derived from the sunlight. Okay, using the energy derived from the light energy. So that is what happens basically in step 4 and 5. Electron transfer through electron transport chain and also the production of ATP. In order to produce ATP at step 5, this one involving just like what we have learned in chapter cellular respiration, there's a chemiosmosis involved. There's a process of establishment of proton gradient, and also chemiosmosis occur in step 5. Okay, chemiosmosis, establishment of proton gradient together with chemiosmosis will result in the production of ATP. The concept is just like what we have learned in the chemiosmotic theory of the cellular respiration topic. Okay. So which photoexcited electron passes from the primary electron acceptor of PS2 to PS1 via an electron transport chain. This electron transport chain made up of plastoquinone, or we say PQ, cytochrome complex, and also a protein called plastocyanine, or written as PC. Okay, that's what happened in step 4. Okay, how is it photophosphorylation will occur? So in step 5, ATP synthesis, okay, ATP will be produced here. So the exergonic flow of electrons to a lower energy level provides energy for the synthesis of ATP. So what happens is that just like in what we learned in cellular respiration, once the electron is being transferred from one electron to another, from one electron carrier to another, energy will be released. That's why it's called exergonic flow. Exergonic flow here means a process that release energy. Okay, the process that release energy. That's what it means by exergonic. Exergonic means release energy. So when the electron is being transferred, there's an energy release. And that energy release from the electron transfer used to pump proton. Okay, used to pump proton. And as the result of the proton pumping, that will establish a proton gradient. Okay, and then that proton gradient will then causing the proton will then support chemiosmosis, where the proton will flow, and when the proton flow through the ATP synthase enzyme, as what we have learned. So the proton will flow through the ATP synthase enzyme, and that ATP synthase will catalyze the synthesis of ATP, just like what we have learned in topic 5, cellular respiration. So as electrons pass through the cytochrome complex, proton are pumped. Okay, proton are pumped from, you guys remember from where? From stroma into the thylakoid space. Okay, from stroma into the thylakoid space, contribute to the proton gradient that is subsequently used in chemiosmosis. Okay, stroma into the thylakoid space. If you guys remember for the cellular respiration, the proton pump from the mitochondrial matrix into the intermembrane space, isn't it? So this one for the photosynthesis, it pump from the you guys just change for the cellular respiration, pump from the mitochondrial matrix, but for the photosynthesis, it pump from the stroma, also the fluid, isn't it? Okay, and then into the thylakoid space. For the cellular respiration, the proton pump from the mitochondrial matrix into the intermembrane space, isn't it? For the photosynthesis, from the stroma, which is the fluid, into the space as well, but not intermembrane space, but thylakoid space. For the photosynthesis. So you guys just change. Okay, and then there's a same like the cellular respiration topic. There's a establishment of the proton gradient. Okay, and then just remember proton gradient, one region higher proton concentration, another region lower proton concentration. So then chemiosmosis will occur. What is chemiosmosis? Remember, is the flow of proton, isn't it? Chemiosmosis is the flow of proton from higher concentration region to lower concentration region. Okay, so this following its concentration gradient is a passive transport. So this proton will flow back, okay, but they will flow through an ATP synthase enzyme, isn't it? So they will flow, this proton will flow through the ATP synthase enzyme. And when the proton flow through this ATP synthase enzyme, then the ATP synthase will use the energy to power ATP synthesis. That energy will be used to phosphorylate ADP into ATP. All right, so this is the overall things you see here. First of all, let's look at the location first. You see, this one is stroma. Okay, stroma having low proton concentration. Okay, and then at the space here inside, this is the whole thing is a thylakoid. This is the whole thing is one thylakoid. We have the thylakoid membrane here, where the thylakoid membrane having the photosystem. You guys see the photosystem here, and then in the thylakoid we have the thylakoid space. Okay, and then outside of the thylakoid is the stroma, is the fluid, isn't it? Okay, so you see the gold arrow. You see the gold arrow first. You see here the gold arrow. The gold arrow shows the electron being transferred. Okay, the electron here from water refill into the P6AO plus, and then the electron transferred to the plastoquinone, and then the electron transferred to the cytochrome complex, and then transferred to plastocyanine. So you see, can you see the red arrow here? The red arrow here. So you see, when the electrons pass through the cytochrome complex, okay, when the electron pass through the cytochrome complex, you see here, the cytochrome complex will pump proton. Pump proton from where? You see this red arrow showing from where to where? The red arrow showing the proton pump from the stroma into the thylakoid space. When we say pump means active transport, means the proton move against concentration gradient. So you see the stroma have low proton concentration and the thylakoid space have high proton concentration. That means the proton is pumped against its concentration gradient, isn't it? So as the electron transfer from plastoquinone to cytochrome complex, the cytochrome complex will pump proton from the stroma into the thylakoid space. Okay, that's one thing. Okay, second thing, from this thylakoid space having high proton concentration, the stroma having low proton concentration, so there's a proton gradient established across this membrane. There's a proton gradient established across the membrane where the thylakoid space having higher proton concentration compared to the stroma having lower proton concentration. So that's a proton gradient here. So because of this proton gradient, that will then support the chemiosmosis. What is chemiosmosis? The flow of proton. The flow of proton back from the thylakoid space back into, you see this dash arrow of red arrow. So you see the proton will then flow back into the stroma from the high proton concentration region of thylakoid space back into the lower proton concentration of stroma. But when they cross back into the stroma, they will cross through the ATP synthase in the protein channel of the ATP synthase. This one is ATP synthase. And then you can see here through this chemiosmosis, ATP is produced, just like what we learned in cellular respiration. So the proton flow through this ATP synthase, and then you see here ADP, okay, added with phosphate group, isn't it? So ADP added with the phosphate group, then we got the ATP here. So this is what we say photophosphorylation. Phosphorylation because we are is the formation of ATP. Why photo? Because at the earlier of the process, we are using the light energy as the source of energy. That's why the whole thing is called photophosphorylation. Okay, all right. That's all about the process occurs in the electron transport chain and then the production of ATP. Now you see what happened. Electron just now is transferred from the plastocyanin to the cytochrome complex, and then it goes to the plastocyanin, isn't it? Then where the plastocyanin will then transfer its electron. So this plastocyanin will then pass the electron to the next photosystem, which is photosystem 1. Just now the electron comes from the photosystem 2, isn't it? So now the electrons will be passed to photosystem 1 by the plastocyanin. Okay, we go to the next step. Okay, before we talk about that, okay, meanwhile, light energy was transferred via light harvesting complex pigments to the PS1 reaction center complex, exciting an electron of the P700 pair of chlorophyll A molecules. Okay, you see here, we are now talking about PS2 now. We have done talking about we are talking about PS1 now. We have done talking about PS2 and the event occurs in the electron transport chain. So now we are talking about the PS1. You see, the same thing happened in photosystem 1. You see there's a light energy here. There's a photon of light striking the pigment molecules in the PS1. So again, you see the black arrow here. The black arrow here show the energy transfer from one pigment molecule to another. Then the energy transferred to here, 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 and then at the end, the energy will be transferred to the P700 molecule, which is the reaction center chlorophyll A in the reaction center complex of the PS1. It's called P700. So when the energy reach the P700, that will cause again the electrons of the P700 to raise to higher energy state or higher energy level. So the electrons of the P700 will be raised, but it does not drop back. Instead, it is being accepted by primary electron acceptor. Okay, so when P700 missing its electron, so P700 will become P700 plus, or will be oxidized to P700 plus. Okay, when P700 oxidized to P700 plus, that means there's an electron hole, isn't it? There's an electron hole that needs to be refilled. That needs to be the electrons of the P700 plus that is missing needs to be replaced. Okay, how the P700 plus able to replace its electrons? So the electron comes from the plastocyanin. Okay, comes from the plastocyanin, where the electrons actually originates from the electrons of the PS2, isn't it? So the plastocyanin here will pass the electrons to the P700 plus. Okay, and then the P700 plus, upon receiving the electrons, then it will be reduced back to P700 and then get ready to receive another energy transfer. Okay, so that's what happened. So the photoexcited electron then transferred to PS1's primary electron acceptor. Okay, this one, the electron from the P700 will be raised to higher energy state and then being accepted by the primary electron acceptor. So this creates an electron hole in the P700, which now called P700 plus. Okay, remember when you see the word P700 plus, that means it is oxidized, it already missing electron. So we need to replace the electron in this P700 plus, so the P700 will accept an electron that reaches the bottom of the ETC from PS2 via the plastocyanin. Okay, the plastocyanin is the one that will pass the electron to the P700 plus, and then P700 plus will then reduce back to P700. Okay, settled. Then what happened to the electron received by the primary electron acceptor? Okay, you see, so that's the next event occur is we have the next electron transport chain, but this electron transport chain only got one member, which is FD. FD stands for ferredoxin. So this primary electron acceptor will pass the electron to the ferredoxin, the member of the second electron transport chain. Okay, and then the ferredoxin then will pass the electron to the NADP plus. Alright. So the photoexcited electrons are passed from the primary electron acceptor of PS1 down a second electron transport chain, okay, where this only consists of one member, which is the ferredoxine of RFD, okay, where ferredoxine is the iron-containing protein. Okay, then you see this diagram. What the where the ferredoxine will pass the electron then. Okay, so next one is step 8. You see the ferredoxine will pass the electrons to NADP plus. Okay, will pass the electrons to NADP plus, catalyzed by enzyme NADP plus reductase. So NADP plus receive the electrons, it receives 2 electrons and will be reduced to NADPH. Okay, NADPH, this molecule is just like the NAD plus and NAD plus and NADH that we learn in cellular respiration topic. It's the same thing. It's an coenzyme, act as a electron acceptor, okay, NAD plus or NADP plus. Both are having the same function, which is accept electrons. Okay, accept electrons, but it's just that NADP plus, they have extra phosphate group. Okay, that's why it's called NADP plus in compared to the NAD plus. NAD plus don't have phosphate group, but NADP plus, they have phosphate group. But they have the same function, which is NADP plus accepting electrons and then reduced to NADPH. So then NADPH will be the electron carrier and carries the electrons and supply the electrons to the Calvin cycle, which is the second part of the photosynthesis. All right. Yeah. Okay, for this second electron transport chain, there's no production of ATP. ATP only produced at the first electron transport chain just now. Okay, so the enzyme, this one is important. You guys have to remember the name of this enzyme. The NADP plus reductase catalyze the transfer of electrons from the ferredoxine to who? To the NADP plus. Okay, then NADP plus will accept 2 electrons and also unite with a proton and become reduced to NADPH, which is then released into the stroma. Okay, so you see the NADP plus here will receive electrons and it will become NADPH. All right. Okay, so that's all about the light reactions. Remember, in light reaction, we have 2 products in light reaction, which are the ATP and also the NADPH. So that's why we say in light reactions, light energy converted into chemical energy in the form of 2 compounds. What are the compounds? ATP and NADPH. And then once we got the ATP and the NADPH, this ATP and NADPH will then supply the energy to the Calvin cycle. ATP will supply chemical energy, NADPH will supply and will be act as reducing power to the Calvin cycle, which is the synthesis part of the photosynthesis. Okay, so in light reaction, remember, we are not yet producing any sugar. Okay, in light reaction, we are not yet producing any sugar. In this light reaction occurring in the thylakoid membrane, only produce the ATP and also the NADPH, and besides that, the byproduct of light reactions also is an oxygen molecule because involve the photolysis of water, where the water is required in these light reactions. Okay, so settle. Now let's try to answer this question, everyone. On your mic and try to answer this question. Okay, we are going to refresh about the process of non-cyclic photophosphorylation. Okay, try to answer this. Step number 1,
photon.
Okay, good. A photon of light strike a pigment molecule in light harvesting complex of
PS2.
PS2. Alright. Boosting one of its
Electron.
Energy.
electron or energy?
Energy.
So higher level, what? Energy ke electron?
Electron.
Electron. Okay, boosting one of its electron to a higher
Energy level.
higher energy level. Higher energy level. Okay, as this electron fall back to its ground state, an electron in a
Nearest pigment.
near
Or nearby pigment.
nearby pigment molecule is simultaneously raised to an
Excited state.
excited state. Okay. Next, the process continue with the what being relate? What being relate? Yeah.
Energy.
Energy. Okay. Energy is being related to other pigment until it reach the
680.
P6.
80.
Okay, P680 P680 pair of chlorophyll A. Okay, then the energy excite and
Electron.
electrons in the P680 pair of chlorophyll A molecule to a
Higher energy.
higher energy state. This energized electron is transferred from the from the
P680.
P680. Then P680 missing electron referred to as
P680 plus.
680 plus. Alright, correct. Next. Okay, how is the electron that have been related to the primary electrons that replace this occur true?
Photolysis of water. Photolysis of water.
Photolysis of water, correct? So photolysis of water, an enzyme catalyze the splitting of a
Water.
water molecule into
Two electrons.
two electrons, two protons, and an
Oxygen atom.
oxygen atom. Right. The electrons from the water molecule splitting are transferred to the
P680.
P680 plus. P680 plus or P680?
Plus.
Plus, right? P680 plus. Each electron replacing one transferred to the primary electron receptor. The electron flow of P680 plus
Few.
is filled. Okay. The
Oxygen.
oxygen atom combined with an oxygen atom generated by the splitting of another water molecule forming
Oxygen
forming oxygen molecule. Okay, good. Next one. Which photoexcited electron passes from the primary electron acceptor of PS2 to PS?
1.
PS1 via ETC. Okay, the ETC is made up of the electron carrier what?
Plastoquinone.
Plastoquinone, a
Cytochrome.
complex, and a protein called plastocyanine. Alright. Okay, then ATP synthesis. The exergonic flow of electrons to a lower energy level provides energy for the synthesis of
ATP.
ATP. Okay, correct. ATP. Okay, as electron pass through the
Cytochrome.
cytochrome complex, proton are pumped from the
Stroma.
into the thylakoid space, contribute to the what gradient?
Proton.
Proton gradient. That is subsequently used in
Chemiosmosis.
chemiosmosis. Okay, meanwhile, light energy was transferred via the light harvesting complex pigments to the PS1 reaction center complex accepting an electron of the
P700.
P700. Okay, the photoaccepted electron then transferred to PS1 primary electron acceptor. Primary electron
Acceptor.
acceptor. So this create an electron
Hole.
Hole.
hole in the P700. The P700 now is called
P700 plus.
P700 plus. Okay. P700 plus accept an electron that reached the bottom of the ATC from PS2,
2.
right? The photoaccepted electrons are passed from the primary electron acceptor of PS1 down a second
ETC.
ATC through the ion containing protein called ferredoxin. This change does not produce ATP. Okay, the enzyme, what is the name of the enzyme? ATP plus? Reductase. Catalyze the transfer of electrons from ferredoxin to
NADPH.
NADP
Plus.
NADP plus. Okay, NADP plus accept 2 electrons and unite with a proton and become reduced to NADPH, which is then released into the stroma. Okay, in light reactions, the in light reaction, light energy is converted into chemical energy in the form of two compounds. What are the form of two compounds?
ATP.
And
NADPH.
NADPH, which supply chemical energy and reducing power to the Kelvin cycle. Okay, thank you very much, everyone.